Let's go on with the next chapter, which will be about the meiotic behavior of these polyploids and the genetic consequences of it. We'll talk now mostly about prophase and metaphase 1 and also anaphase 1. As a reminder, what happens during those phases is that chromosomes pair together 2 by 2, and this pairing is very important for the next step, which is the separation of them and the segregation of one chromosome to one pole of the cell and the segregation of the other to the other pole. If the pairing doesn't happen properly, the segregation won't happen properly either. Which leads to the problems that happen in polyploids. One main major problem of polyploids is that they are sterile, or to a certain extent. If we look at triploids, the first problem is, of course, 3 is an uneven number, which means that dividing this by 2 is just impossible. But an extra problem that triploids face is that during metaphase 1, so during the chromosome pairing, you can have the formation of trivalent, so the 3 chromosomes will form a triplex together, or you can have formation of a bivalent, and then the last chromosome is left alone. In these cases, so trivalent or univalent, the pairing is not proper. If the pairing is not proper, the segregation of those chromosomes won't work either. So in the case of the trivalent and univalent, you're probably gonna start to have like random segregation. So chromosome can go in one cell or the other and in a very random fashion. If this happens, it leads to the risk of having aneuploid gametes, which means that certain chromosomes will be in extra copy, while others will be in less number of copies. And if gametes are aneuploid, the result is very often that they are non-functional. They are lethal. Okay. If we go now, if we think of autotetraploids, here we have an even number, so in theory we could have just pairing and then dividing by 2 and things would be fine. This can indeed happen, so let's say you can have 2 chromosomes out of the 4 that match together, then they will form a proper pairing, divide properly, and everything will be fine. It can actually also work fine in terms of quadrivalent, so when you have the 4 of them going together, pairing together, because these can still be divided by 2, and then you will have 2 in one daughter cell and the 2 other in the other cell. But we can also have the case of one chromosome being left alone because the 3 others will form a trivalent, as it's the case for the triploids. And in this case, again, we're going to have the problem of random segregation of those chromosomes, which can lead to aneuploid gametes and being non-functional. And what you have to consider is that in any plant species, there is more than one chromosome. It's so often 5, 6 or more chromosomes. So to have a proper gamete, it means that this scenario needs to happen for all the chromosomes. If only for one chromosome you have this scenario happening, then probably the gamete will be non-functional. So the probability to get functional gametes becomes as low as the number of chromosomes that exist in a plant. And in the case of allopolyploids, all this is valid. You have the same kind of problems, but on top of that, you have another problem. The other problem is that this probably won't happen properly because, as we discussed before, allopolyploids come from different species, and these chromosomes from one species are the homologous chromosome from the other species. The duplex between these homologous chromosomes won't be proper, and as I told you again, the pairing is sequence-based, and the pairing is very important for the segregation of the chromosomes. So again, if you have a problem in duplexing the chromosomes, you have the risk to get uni or trivalent, of course, and you will have again random segregation of the chromosomes, leading again to aneuploid gametes. Let's take an example. Here is a brassica allopolyploid, and we are in anaphase 1. So anaphase 1 is already after the chromosome pairing and after the chromosome already started to migrate from one pole or to the other. Okay? And what you have here is actually some chromosomes that probably couldn't pair properly during metaphase 1. Because as you can see, they are lagging behind because of the problem we discussed. So these two chromosomes can randomly end up in like the future cell here after cytokinesis, or maybe in this cell in a very random fashion, and that can lead to aneuploidy. And if we look more carefully what's happening in the same example, now we are in metaphase 1, so during the time where chromosomes pair together. The technique that has been used is very old but also very useful. It's called genomic in situ hybridization. The principle is very simple. You just have an antibodies that have a fluorophore, so they fluoresce either green or red, and you can direct those antibodies to a specific sequence, and you can choose sequence that is specific to one subgenome or to the other. Okay, so for example, here Brassica has two different genome because it's an allopolyploid from two different species, so species A, species B. And what you can see here, for example, is pairing of chromosomes of the genome A, genome B. What happens often when we try to create new polyploid is that some lines, so after creating these polyploids you have the progeny, some of the progeny will actually be fully fertile, but some won't. And in the case here, for example, it's a line which has no fertility problems. And what you can see, what is quite interesting, is actually the chromosome from one genome, here, here, actually will pair together. And here, for the green, so genome B, they will also pair together. You won't find any duplex or triplex or whatever between homologous chromosomes. If you don't have any pairing between homologous chromosomes, it means the duplex will work properly. But now let's take another line of the same allopolyploid that is actually partially sterile. What you can see that happens is that in this case here, it's very clear, but you can also see it here, is that you're gonna have formation of multiplexes between homologous, so between chromosome of genome A and genome B. This multiplex is probably not perfect, which means not perfect pairing, means not perfect segregation of the chromosome, which means aneuploidy and sterility. But that's not it. On top of sterility, we're gonna now look at another problem that polyploid faces. So let's take this example of Trachopogon species. Actually, there are two endemic species of Europe, Trachopodon pradensis and Trachopogon dubius. One more like Northern Europe and the other more like Southern Europe. In the early 90s, what happened with human action, those two were imported to America, and they grew in the same environment basically, and they were used for ornamental species. What happened there is that actually they hybridized and they formed a new allopolyploid species. This happened only 80 years ago, so it's an extremely recent event. And that leads to this allopolyploid of its allotetraploid to N4X equal 24. So the authors here sampled individuals of this new allopolyploid species in different places. As you can see here, each spot is a population they sampled. Okay, and what they did, they studied the karyotype of these individuals. Karyotype means that you just stain the chromosomes and you align them together and you can see how many they are and so on and so forth. So here are the karyotypes of the parental species. There again they used genomic in situ hybridization to stain like one species in green, the other in orange. And as a reminder, here are two homologous chromosomes versus here two homologous chromosomes. Okay, and let's say that if everything is fine in the allopolyploids, that's a theoretical expectation. So if chromosome pairing and everything works together, you should find this as allopolyploids. Okay, that's not exactly what they found. We have here three different individuals of one population only, and plenty of problems seem to arise. So for example here, if we take the chromosome E, you see that for one species genome in the allopolyploid you have 4 chromosomes, while for the other species genome all the chromosomes are lost. Another problem is like what you have the arrow here, you have some green here and some orange there. That is a phenomenon of translocation, which means you have some recombination that happened between the two, because probably the sequence here is quite similar, and then that will lead to this hybrid chromosome. So pieces of a chromosome of the species chromosome until here, and then the other part is from another species. Now if we look at the individual two, you can see that the karyotype is extremely different. So that's very unexpected, you know, because if you, all of us in this course, for example, we probably all have the same karyotype. It's like 99% chances. But this is not the case. As you can see, the karyotype is very different. For example, in chromosome B and C, you have only 1 chromosome from one subgenome, but you have 3 in the other subgenome. And again, some translocation events here. And if we look at the third, you got it, it's even different again, like missing all the chromosome, 4 here, translocation here quite big, fragment, and here as well. Okay, so now if we look at the summary of what they found for all the population, they looked at the euploid. So euploid means that you have the proper number of chromosomes. So basically you have this scenario, 2 from each species, nothing special. And what you can see is that this is not the majority, far from it. The majority is instead what they call compensated aneuploidy. So that means that you have aneuploidy if you look at one subgenome. So here you have 2 extra copy or 1 single chromosome, but it's actually compensated by the fact that in the other subgenome you have absolutely no chromosome. Okay, so that's what they call compensated aneuploidy. While you have numerical aneuploidy, which means true aneuploidy. So you don't have a total of 4 chromosomes, but you have more sometimes. Okay, and as a conclusion, this compensated aneuploidy phenomenon is extremely frequent, as you can see, and chromosome translocation is also very frequent. And something extremely interesting is that the karyotypes are not fixed, so they are not the same even within one population. And that's the main conclusion, is that you have high genomic instability on the short term for polyploids. And it's quite a problem, maybe not in the wild, but a problem for breeding, and we'll talk a bit more about it later. But now the other question that we can wonder, okay, that happens after a very few decades or maybe even 100 years, but what about a more evolutionary long term, like 1,000 years or hundreds of thousand years? And we will study some cases during the class for that. Okay, so as a conclusion, here is a little quiz. The first question I ask you is, what is the main cause of sterility in polyploids? This is an open question. Then, why does this happen? What is the problem exactly? Then, when does this happen? And finally, the same problem as other consequences, and which one?